Well, we've heard about new hormonal therapies. We've heard about exciting and unexpectedly positive results with the radionuclide. Now I think it's time to hear about the role of inflammatory cytokines and their potential as a therapeutic target for treatment of prostate cancer. Our speaker is David Waugh, who is from Queen's University in Belfast.
Okay, thank you, Dr. Chabner, and thank you to STO for the invitation to speak to you this afternoon. I'm going to focus very much on work that's been generated by my own lab in discussing the importance really of inflammation as an aspect, a very important aspect of tumor biology in prostate cancer. And really in keeping with the translational emphasis of the meeting, I'm going to talk about really inflammatory chemokines, but really in the second part of the talk, start to begin to elucidate where, what, and when we should be targeting this inflammatory process in prostate cancer and the opportunities that the data that's emerging from my lab might give us in tackling this very significant aspect of the disease. So, as I've mentioned, inflammation is a well-established hallmark of many cancers. And it's sort of comforting in a way that despite the pathology being recognized for inflammation in disease for a long time, it's now only as we've begun to understand the molecular basis of that inflammation and the role of cytokines and chemokines that tumor-promoting inflammation has been added in the updated Hanahan and Weinberg review. And this tumor-promoting inflammation is now characterized as an enabling characteristic of cancer, really because these pro-inflammatory chemokines and cytokines can sustain many of these established hallmarks of cancer. And inflammation has a very prominent role in prostate cancer. Indeed, there's even been a pathology that's been attributed to the role of inflammation in this disease. And this has been led by Angelo DiMarzo and his colleagues at Johns Hopkins University in Baltimore, where they've shown that chronic and repetitious injury drives this damage-induced proliferation of the inflammatory cell infiltration into the tumor site, leading to these zones of focal epithelial cell atrophy. And this injury is spurred on by insults such as bacterial and viral insults, changes in hormonal balance, dietary factors, urine reflux, for example. And this all drives this innate immune response. And as a result of this inflammation and this gross gain in proliferative rate, the risk of cancer development is associated and arises from this, which gives rise to this increased risk of acquiring the gene mutations that sustain cancer, the damage to the DNA that comes from the inflammatory byproducts, so you get an awful lot of oxidative stress, localized nitric oxide release, which can directly damage the DNA bases, and also because of a result of the activated stromal response, which can then sustain that rate of cell proliferation. So, in essence, the aspect here of this prostatic inflammatory atrophy is that the tumor-promoting inflammation gives rise to the genome instability and mutation. And this pathology sits here as one of the key pre-malignant lesions that now is being examined in many, many studies. And this gives rise then to the subsequent progression to this prostatic intraepithelial neoplasia known as PIN, which then, under certain genetic conditions, will give rise to the invasive carcinoma. Now, my lab has taken a slightly different and alternate approach, typical of the Irish way, but that's really to look at whether genome instability and mutation actually can sustain and give rise to the tumor-promoting inflammation itself. And of course, we've looked at inflammatory chemokines. This is the focus of my lab. Chemokines are an established entity of the immune response. They drive the influx of cells into sites of injury and inflammation in a chemotactic migration factor, which is stimulated by the localized release of the ligand, stimulating the cells that express the receptor to migrate up that concentration gradient. And as I say, although classically associated with the immune system, there's now a very firmly established role for these chemokines in mediating tumor stromal communication in many cancers. Now, in prostate cancer, there are three primary chemokine signaling axes that have been established. Ken Pienta at Michigan has led the development of, or characterization of the importance of CCL2, binding to its receptor CCR2. Russell Takeman, also at Michigan, has been heavily involved in the characterization of CXCL12 signaling through its receptor CXCR4 in prostate cancer. And my lab and many others from MD Anderson have established the role of interleukin-8 in signaling through its cognate receptors and its relevance to the biology of prostate cancer. Now, in terms of the gene instability in prostate cancer, we've already heard Johan talk about the molecular stratification. These are the classical tumor suppressor genes which are lost early on in the onset of prostate cancer. So loss of P27, gain of this homeobox, or loss of this homeobox, NKX 3.1, this classical phosphatase P10, and indeed even P53. And this coupled with these oncogenic gains, such as MYC, ERG, ETS transcription factors, and indeed the androgen receptor itself, give rise to that invasive and progressive disease. Now, PTEN is the most frequently mutated gene in prostate cancer. Its expression is also frequently downregulated as a result of promoter methylation. And this downregulation or mutation is frequently detected in these pre-malignant lesions that I've already talked about, the PIA and the PIN foci. Many patients already present with loss of one allele, and if you look at advanced cases, there's frequent downregulation or loss of both alleles of this classic phosphatase. And a lot of very elegant mouse models have shown that loss of PTEN, be it in the haploinsufficient condition or indeed complete loss of PTEN, gives rise to a pathology that recapitulates human disease. In other words, you get the arising phenotype of these pre-malignant lesions, and then coupled with these other oncogenic gains or loss of other tumor suppressors, you get that progression to invasive carcinoma. And this is work that's really stemmed from Pier Paolo Pandolfi and Corey Abate-Shen's lab. So our hypothesis has been to really look at what would be the impact of PTEN loss and this genetic instability that arises from this aberration on the inflammation, and looking particularly at the inflammatory chemokine signaling. And so we've taken a range of cell lines in the lab, well-characterized and established models of prostate cancer, which reflect both the androgen-dependent and their androgen-independent status. And you can see that we have a mixed population here of cells that still retain expression of PTEN. Now, we've used transient sRNAs in the first instance to downregulate expression of PTEN in the DU145 and 22RV1 cells. And you can see that there's a very, very, very selective regulation of the chemokine signaling pathways when we silence PTEN. So here, in both cases, we get a selective induction of CXCL8 or interleukin-8, but we don't see any impact of PTEN loss upon the other chemokines implicated in prostate cancer. So there's no change in CCL2, no change in CXCL12. And when we look at the corresponding regulation of the receptors that mediate the biological activity of these chemokines, again, we see a very selective induction of CXCR2, which mediates the signal for CXCL8, but at the receptors that mediate the effects of the other chemokines are not affected by PTEN status. We've also backed that up looking using a stable SH-mediated downregulation of PTEN in the DU145 cells. So you can see here we have a range of clones in which we see a 50% downregulation of PTEN's expression consistent with a haploinsufficient condition, and a complete loss of PTEN expression. And again, you can see that irrespective, if you've just knocked down our haploinsufficient conditions, you see this marked increase in CXCL8 expression, but again, the loss of PTEN has no effect on the expression of these other chemokines, befitting the selective upregulation we've seen before. And you can see here that upon loss, or increasing loss of PTEN, you see an increasing gain in the secretion of CXCL8 from these cells. Now, we think that the fact that there's a disconnect between message and secretion here reflects that the loss of PTEN is also impacting on the increased efficacy of translation in these cells, an increased synthesis of IL-8 or CXCL8, and therefore an increased secretion. And again, if you look at the receptor status, here we're comparing the haploinsufficient condition, and you can again see that gain in CXCR2 cell surface expression upon PTEN loss. The in vitro data is also substantiated by work we've done looking at the expression of the mouse ortholog chemokine KC in heterozygous PTEN prostate tissue. So this is mouse tissue supplied to us by Chip Albanese at Georgetown University. And you can see that as we move from normal epithelium in the heterozygous condition across this cytological atypia, you can see that there's a really increasing gain in the expression, again, of KC. So the in vivo data here is backing up the in vitro model. Now, in terms of translating this into prostate cancer itself, we published a study several years ago which characterized in a histopathology fashion the over-increased expression of CXCL8 and its receptors consistent with a changing stage of disease. So the expression was greatest in the androgen-independent setting, but we could detect increased expression of CXCL8 and its receptors in those very early pre-malignant pin foci that we examined. Now, we have to go back, obviously, now and do the retrospective validation against PTEN status in this tissue cohort. But importantly, we did see a strong correlation of increased CXCL8 and its receptors with markers of angiogenesis proliferation. So again, it befits that inflammatory proliferative drive that has been established by the DiMarzo group in Johns Hopkins. And there are many other studies, I'm just citing Robert Veltri's work here, which shows that serum levels of IL-8 are detected or increased as you go through stage of disease. So you can even see changes in the serum level when you look at patients with pathologically confirmed localized disease, and that gets even more significant when you look at patients with confirmed metastatic disease. So what does IL-8 do, or CXCL8 do, in the context of the human prostate? This is looking really now at both the autocrine and the paracrine effects of this chemokine. So in terms of the autocrine effects, IL-8 can drive the proliferation of the epithelial cells. We published that several years ago, and we've now subsequently shown that IL-8 can activate and sustain proliferation through induction of the androgen receptor expression and activation in these cells. There's also a very strong literature now that we've developed showing that the synthesis and secretion of IL-8 signaling will sustain chemoresistance to many novel molecular targeted agents in prostate cancer. Work I'll show later on will show the impact of this in terms of survival. In terms of the paracrine effects, then, well, IL-8 is a very potent inducer of neutrophil infiltration and also macrophage infiltration. So you can see here that it's going to have an impact in orchestrating the immune complement within the prostate microenvironment. It will also have effects on fibroblasts, inducing growth factor synthesis, for example. So all of these would befit, if you look at the recent data that's come from Jim Quigley's lab at Scripps, that the signaling of IL-8 to neutrophils can sustain MMP9 synthesis, and this also then sustains the angiogenic response that's been attributed by many years of work by Rob Streeter at UCLA to this particular chemokine. So in terms then of the translational impact, what does this research tell us? Well, if we look at prostate cancer trials that are currently being conducted or have been conducted, really targeting chemokine signaling has been restricted almost exclusively to the CCL2 and CCR2 axis. So there have been antibodies that have been developed by Centocor and also by Millennium that target this signaling axis. And there's good justification for why this is so, because this signaling axis pays particular attention or importance in the context of metastatic disease within the bone, where CCL2 and CCR2 signaling regulates the communication between tumor cells and the osteoblasts. But if you look at this targeting, all of this pathway is still left untouched. And we think that the inability and the redundancy of the system and the inability of the current protocols leaving this untouched can essentially function to compensate for targeting this particular pathway. So the data that I've shown you to date shows that under genetic stability of PTEN, instability of PTEN loss, and accentuated by either chemotherapy stress or hypoxia stress, you get an activation of these transcription factors which sustains the increased expression of this ligand and its receptors. So what does IL-8 signaling or CXCL8 signaling do in the context of a prostate cancer cell? This is a figure taken from our clinical cancer research review. And you can see that signaling through these receptors activates many of the transcription factors associated with disease progression in terms of angiogenesis and survival. So here we have activation of HIF, which we've just submitted for publication. There's a strong coupling of IL-8 to the activation of NF-kappa B, AP1, the androgen receptor, which Johan's already talked about. So you can see that this signaling pathway affects many of the aggressive characteristics that prostate cancer metastasis requires. Now, we've shown you data that under PTEN loss, you get this selective increase in CXCL8 signaling in vitro and in vivo. So what's the significance? Well, we're going to show you data that shows that IL-8, induction of this IL-8 is probably a good example of the synthetic lethality or induced essentiality phenotype under PTEN depleted conditions. So this again is taking the DU145 cells, and in this context we've used two siRNA oligonucleotides that target IL-8. And you can see that there's a marked effect in downregulating the production of IL-8 in these cells. Now what we've done is we've taken the PTEN expressing cells and we've transfected them with those two oligonucleotides, and you can see that the viability of the cells really isn't markedly affected. But where we've got the haploinsufficient PTEN status or the complete loss of PTEN under either transfection conditions, now you see a marked decrease in the viability of these cells. We've also examined this in the bone metastasis model, the PC3 cell line, and again, where we've used a short hairpin strategy, these are PTEN null to begin with, where we've used a short hairpin strategy to knock out IL-8 using 4 different sequences. Again, we see a marked decrease in the viability. And we actually had to back off in the transfection conditions to show any gain of these colonies over 10 days. So very effective knockdown resulted in no colony formation at all. Now, in the context of early therapy then, as Joe's already alluded to, radiation therapy is one of the early interventions used for patients who have relapsed either surgery or not eligible for surgery. And again, we've used this PTEN deficient model to demonstrate the impact of PTEN loss upon radiation sensitivity. So here we have the haploinsufficient condition, or 50% expression of PTEN relative to normal, and you can see that the impact of radiotherapy is pushed to the right. And here again, under PTEN completely null conditions, radiation is, the cells are less sensitive to the radiation. Now what we've done is we've taken an siRNA strategy that's co-targeting or co-knocking down both CXCR1 and CXCR2, the receptors that mediate the effects of IL-8 in the tumor cells, and we've irradiated them. And so under these conditions here, this is PTEN expression. CXCR1 and 2 is not having any effect. But under this condition here, where we've got 50% of the normal PTEN level, now the radiation kill curve is sensitized upon the loss of the CXCL8 signaling. And the same is true under the PTEN null condition. So obviously under these conditions, CXCL8 targeting through the receptors is having a dramatic radiation sensitization effect. So where, when, and what to target? Well, I've shown you that inflammation has a very important role in this progression all the way through disease. I've shown you in terms of a molecular stratified approach that if you lose PTEN status, you get this exacerbated increase in and selective increase in the expression of this chemokine and its receptors. And we think that this has a very important multifunctional aspect in terms of promoting disease. So where can we use this information? The information in terms of survival suggests that if we can detect disease early, even at this pre-malignant stage, or even an early locally organ-confined disease, targeting IL-8 signaling could actually induce this sort of synthetic lethality phenotype and halt tumor progression. One of the major questions I think that still remains for us to address is, what's the best strategy here? Should we target the ligand, or should we be going after the 2 receptors? And which of those receptors, or indeed both of those receptors, should we be targeting? And that's really the challenge, I think, from this meeting, is really how do we engage with the pharmaceutical industry to start challenging this? There are antibodies that have been developed by Apgenics that target the ligand. There are antibodies and small molecules which have been developed by pharma now to target CXCR2 receptors, and I think this gives us an opportunity to go after this. And I clearly think that in the context of the current work, targeting IL-8 signaling becomes highly relevant under those PTEN deficient conditions. So this is really an approach where I'm really following up on the comment that Johan's made, where if we know the genetic deficiency or status of the patient, this becomes a relevant strategy to employ. Is targeting one chemokine pathway enough? Well, I've already alluded to the fact that CCL2 and CCR2 have been targeted in clinical trials already, and Johan will maybe talk about the data that's emerged from those trials in the ensuing discussion. But if we look at the effects of paracrine IL-8 signaling, we know that it can affect neutrophil infiltration. We know that dampening its effects would dampen the macrophage infiltration as well. But we know that CCL2 is present in the bone metastases, and CCL2 can have impacts on the macrophage activation and the microenvironment. We also know from data that is just showing up here on the top that if you stimulate fibroblasts with IL-8 or CXCL8, you also induce expression and increased production or synthesis of CCL2 from these cells. So the paracrine effect here is amplifying and diversifying the inflammatory milieu within the microenvironment. And so perhaps, given the redundancy in the systems, yeah, we should be taking strategies that target the tumor-osteoblast interaction around CCR2, but combining that with strategies that would maybe target this CXCL8, CXCR2 signaling axis, particularly under this genetic context. And again, the genetic context here is markedly enriched in bone metastasis or metastatic disease. So in conclusion then, I've shown you that genetic instability selectively upregulates the autocrine and paracrine signaling for CXCL8 over the other chemokines that have previously been implicated in prostate cancer. Autocrine CXCL8 signaling enables the cells to withstand the stress incurred by that genetic instability arising from PTEN loss, and the paracrine CXCL8 signaling contributes to immune cell recruitment and amplification of the inflammatory PTEN deficient tumor microenvironment. And I think this has major consequences, both for angiogenesis and also then for invasive growth. I think the future translational perspectives are that targeting CXCL8 signaling provides an opportunity for us to directly target pro-inflammatory signaling in the early stages of disease, perhaps in combination with radiotherapy, and simultaneously while we're doing so, perturb not just the lethality in the tumor cells, but affect the ability for the tumor cells to modulate stromal response. And I think, again, it clearly has implications in combination approaches with many of the interventions, such as radiotherapy, also hormonal therapies, which we've previously published. So I will finish there and just to acknowledge the collaborators, both local and global, that have helped us with this research program, our funders, Cancer Research UK, and just to acknowledge Pamela Maxwell and Johnny Coulter, whose work I've presented today, assisted by Jess Neeson, a PhD student in the lab. Thank you very much.